Today, the sort of invited guests are both local, and they're both here at Hopkins. And we have Hari Easwaran, and Hari was here. Was it the last meeting or was it the one before? Anyway, Hari gave a nice long rifles. Hari is an associate professor here with a primary appointment in oncology, and he works on cancer epigenetics, and did his PhD work actually on the fundamental mechanism of one of the DNA methylase enzymes. So he's been looking a lot at DNA methylation with respect to cancer, but also very interestingly enough, something many of us are very interested in is the relationship between the changes in the epigenome and that are associated with increased age, with aging. And so, and this will probably tie in, hopefully, something to what Noel's been looking at too, because so Noel has been doing a lot of focus on basically the other two horrible diseases that afflict most of us as we get older, namely cardiovascular disease and metabolic syndromes, and also obesity, which affects many of us as we are younger. And so there's a strong age component, and Elizabeth and I have certainly been looking a lot into this in terms of age and cancer, trying to understand what are sort of the biological mechanisms that underlie the dramatic increases in the disease as we get older. And so there are several of these diseases, and we think, epigenetics must be playing some type of a role here, right? So Noel is relatively new to us, and again, we're trying to sort of cross-pollinate fields. So he's not, as far as I know, a cancer researcher. You're into all kinds of other very interesting things, and the microbiome, which we'll get into, and neonatal microbiome, which I hadn't even really considered. But this idea that, say, for instance, if you have a C-section, you don't get exposed. Well, you don't, but the baby doesn't get exposed necessarily to the mother's microbiome, and maybe it's aberrantly colonized, and it could have effects later in life on things such as levels of obesity and other diseases and things. This is just fascinating. I think it's going to be a really interesting thing to get into. And of course, Cindy Sears is here, and she's big on microbiome, gut microbiome, and cancer. So I think this is going to be great. So Noel is relatively new to us. He's also an associate professor. His primary appointment is in epidemiology. He's over at the Welch Center, and in the divisions of cardiovascular disease and clinical epi. And so Noel, what we do a little bit here before we get going is the other thing that Don really liked to do, because again, he really liked to get to know people. He wanted this to be a little bit different. We don't just read your number of publications and all this kind of stuff. We want to get to know you just a little bit. We're not going to dwell too long, but so we want to know about you, Noel. Where were you born? Where did you grow up? What were your parents doing? And what were you into, say, as a kid in grade school and high school? Were you like a science nerd? Were you a bio nerd? Were you like a sports nerd? You just watch TV all the time? So tell us your brief life history. Where were you born?
Wow, that's cool. I didn't usually get to talk about
Surprise.
where I'm from. So if you couldn't pick up from my accent, I'm from the Midwest, Wisconsin, born and raised, actually rural Wisconsin. So a town of about 8,000 people.
What town?
Burlington, southeast corner of Wisconsin.
My wife's family's from a place called Tigerton. Do you know where that is? I don't know.
I don't know where Tigerton is.
I don't know it that well, but yeah.
So it's close. It's southeast Wisconsin, close enough to Kenosha and the Illinois border. So
okay.
So both of my parents came from Chicago area, and I grew up watching Chicago Bears football with my grandparents. But
what did your parents do?
So my parents, my dad was a stockbroker in Chicago.
Oh, wow.
And my mom was a teacher and worked in inner city Kenosha for a while, then worked in Milwaukee. So Milwaukee, Chicago, splitting the distance was Burlington essentially.
Cool.
And it was nice. It was a nice small community, but got to grow up near some lakes and play a lot outside, very different than how my kids are getting raised right now. We do have a backyard, but that's about as close as they get to playing in the woods like I played in the woods. So I grew up really, I guess, bringing it to the microbiome, being exposed to just outdoors a lot, which was great. And played a little bit of violin growing up. I guess that was something that's interesting. And then played sports. I was a big tennis player and baseball player.
Oh, wow.
But got interested actually in nutrition science. Was something that got me really interested when I went to the University of Minnesota. So from Wisconsin, I went to Minnesota, studied. I was a gopher. Did my undergrad in nutrition science. Worked with Dan Gallagher, who did a lot of basic science stuff in my undergrad. And then when I was graduating, I said, I'm interested in doing graduate studies, but I'm not sure that I want to go right away. It might take a little time. And had a long conversation. And he said, well, if you're interested in more of the multifactorial metabolic diseases, you really might consider going into something like epidemiology, nutritional epidemiology, just because a lot of those multifactorial diseases take, have a huge human element or social element to them. It's not just so biological and single pathway. So I kind of took that bit of suggestion from him, took a year to actually go down to South America and work for a United Nations program. I went down to Bogota, Colombia. It also allowed me to further my Spanish, which I had started during college. And took a year and then applied to graduate school when I was there, came back, did my graduate program at the University of Minnesota, included my master's in public health and then PhD. And then after my PhD, I did a postdoc at Columbia University. The postdoc was a T32 program that married kind of basic science nutrition with population health and more epidemiology. So that really allowed me to get this introduction to integration of the microbiome into longitudinal studies. So I worked with Marty Blazer, his wife, Maria Gloria Dominguez Bello, two of the thought leaders in the microbiome, really kind of started the field. And that's where I got this interest in early life, pioneering bacteria and their role for life course epidemiology, or the adult origins of later disease. So I'll go into more depth about that.
Well, yeah, that's really fascinating. The whole concept of that just blows my mind. So here's a question we always ask people. We'll put you on the spot slightly. What was the title of your thesis?
Determinants and consequences of early puberty.
Oh, okay. That's a pretty straightforward title. You got it. Most people don't get it. And we always raise that, especially for the younger members of the audience, and we kind of point out to them this thing that you're focused on that you think is the most important thing in the universe. Years from now, most of us really we can't recite the exact title of our PhD thesis. But you got it. That's great.
I was close. It may have been nutritional determinants and something like that, but yeah.
Close enough. Cool.
I'm also, a bit younger than maybe some of the people who you bring on. So
great. Okay. So, yeah, so a lot to delve into. I don't know where to launch off into first. It's like microbiome or interactions between microbiome and epigenetics and disease. But I just have one real quick basic question now that you just mentioned that, is that, and I don't know the answer to this, but I assume it's known. What, from epi, like what's known about, say, do we know if there's any association between delivery method and, say, cancer risk later in life, or specific cancers or anything like that? I've never heard of it, but I don't know. Seems like that's something that would have been studied.
Yeah, I think that's a really good question. I know that the reason it probably hasn't been rigorously studied is because it requires longitudinal cohorts.
Yeah, that's true.
Or you rely on self-report of delivery mode, which can be done. There is a group at Harvard who's looking at the offspring of the nurses from the Nurses Health Study, and it's called the GUT study, the Growing Up Together study. And they've looked now in the sibling designs or within family designs, of one of the stronger designs in epidemiology, at the discordance between sibling pairs that one was delivered by vaginal delivery, one by C-section delivery, and have found that those that were delivered by C-section delivery have higher risk of obesity, both childhood, adolescence, and later life, and type 2 diabetes. And this is published in like JAMA 2020. But I haven't seen them look at cancer. I'm not sure if that's because of sample size. They probably don't have sufficient sample size for many cancers, but maybe that's something that's on the horizon. And maybe, larger Scandinavian cohorts could do something like that.
Yeah, that's a good idea.
Can I jump in too?
Of course.
This is
you're
the
supposed
jumping
to.
around.
Everybody's
This is the
supposed to
jumping
jump in.
around. Yeah. So, in the old days, there was a correlation between C-section and birth-related things like the size of the baby, right? So the baby's stuck, can't get out, right? So there are studies in Scandinavia. So that would be a potential confounder,
So
right?
you're saying like reversing cause and effect a little bit. So the babies needs a C-section because those would tend to be babies that are bigger, or they.
In the old days, there were reasons, right? And that was one of the reasons, right? Larger size baby and larger size baby. There are studies from Scandinavia trying to relate big birth size to risk of cancer. Certainly small babies have a higher risk of cardiovascular disease because of smaller vessels. So that's part of the Barker hypothesis, right? So these in utero determinants of small for gestational age, higher risk of cardiovascular disease. For cancer, it's not been that clear whether or not bigger babies actually have a higher risk of cancer later in life, but it has been studied. But again, if you're trying to study with long enough follow-up, there are going to be correlations between vaginal versus C-section and the size of the baby and other problems that would dictate who has a C-section versus who doesn't. In the modern world, sometimes they're scheduled for convenience, and so that might disconnect some of the birth-related features from the need for C-section. So maybe in the future there'd be some research that could relate.
But wouldn't you have birth weight
Yeah.
data that you could then, factor in? Yeah, adjust.
Yeah,
Yeah,
but
we
I would imagine there's still reasons, right? It's like a confounding by indication, and so there'd have to be like more complex analysis than just adjusting for birth weight. Anyway, go ahead, Noel, your turn.
Oh, I was going to say, Elizabeth.
Yeah.
I get this in talks, and I didn't know we were going to talk about C-section so much because I do other stuff too. But
yeah.
So as you mentioned, like that's like the number one thing we put in our model after age and sex. And there could be residual confounding. And these are birth cohort studies where we actually measured length and weight of the baby at birth. So hopefully there's not a ton of residual confounding by that particular factor. But there's also catch-up growth for low birth weight babies, high-risk pregnancies. So we try to do our best to account for that with model adjustment, which is, not perfect. But then what is the next thing we could do to get at causality, right? Well, you could do experiments in mice. So Maria Gloria, my mentor, is at Rutgers. She's done a mouse experiment looking at C-section versus vaginal delivered mice. And C-section delivered mice also accumulate more body fat over the first 32 weeks of their life. So that's interesting. And then we said, well, how can we test this in humans? Because we can't randomize humans to C-section or vaginal delivery. But what you can do is you can take one component, which is the sharing of microbes from the mom to the baby, and randomize that. So that's what we're trying to do in this pilot study, to say, do maternal microbes in some way confer a protective or have some protection for the baby for metabolic diseases, maybe other outcomes also. So we've submitted our abstract to the Pediatric Academic Society. We'll be presenting that in April. So that's kind of
question here. Even before birth, do we know if fetuses are exposed to bacteria in I just saw that this is still debated right now, whether the amniotic fluid and the environment where the fetuses are microbe free or they have some bacteria. Is there any
Thanks for the question, Hassan. This is a pretty hotly debated topic in microbiology, and it started getting more hot in 2014 because there was a paper from Kirsty Agard, who's an OBGYN, and she published on the presence of a placental microbiome. And when you say microbiome, that suggests that it's a living microbiome. And there was a lot of pushback because for years, the theory was that it's a sterile in utero environment, but this was suggesting that there's some sharing of bacteria from mom to baby. So after that 2014 study, a lot of people started to look at the methods from collection of the placenta samples and other amniotic fluid samples and then also the pipeline and the processing of the samples and sequencing. And really, there's been several studies who have kind of refuted that evidence, suggesting that much of the placenta microbiome was either contamination from the sequencing or artificial, like bacterial remnants after a phagocytic kind of a fight going on, and it was probably not live DNA if there was some low biomass DNA there. So that's just to say that I think the consensus from my perspective is that there's probably still not a lot of in utero bacteria, and the primary sharing of bacteria comes at birth.
So in that same line, what is the wisdom right now about the bacteria in the milk, in breastfeeding milk? Where does that originate from?
Another good question. So I think the theory now is that those are real bacteria that are being shared with the baby. Unknown where they're coming from. There's some concordance between the maternal gut bacteria and the bacteria in her breast milk. But there's also, I don't think it's been completely disentangled from the baby's mouth, the bacteria in the baby's mouth colonizing the breast, and then there's being in the collection that also being part of the bacteria that you find in the breast milk. But somebody who you might consider reading up on, Megan Azad, who's at University of Manitoba, is doing a lot of work right now in the breast milk microbiome. And unlike, for example, meconium, which is the first discharge from the baby at birth, breast milk consistently has bacteria in it. So it's not as low biomass as the in utero samples I was just talking about. I think it's more likely that it's a real microbiome.
And since you said meconium, was there any microbiome studies done on meconium to see what are those bugs in that first waste?
Yes, we've looked at that. It's the thing that concerns me is that, again, there could be contamination, and some meconium has bugs and others doesn't. So it makes me feel like those that have some microorganisms present, it may just be due to sloppy collection of the meconium. So I think that it suggests that there's probably not a lot of in utero sharing, and you can't find a lot in meconium, but others have found it and published on it. So
thank you.
With respect to cancer, I know I've seen maybe 1 or 2 talks, I think, through the Cancer Center over the last year or so, where people have presented the idea that basically, as far as I could tell from some of them, it seemed like they're saying that there are actual bacteria like almost everywhere in our body, in all the organs, regardless of whether or not they have a direct communication, to the external world. So I don't know. I've never known what to make of that, whether it's, like you said, just contamination, and they're probably using PCR-based assays. And maybe Cindy could chime in here. She might know more about that too, but it seems to have important potential implications if it's true, but I don't know how true do you think that is, and does my brain have bacteria in it?
Cindy might comment on how germ-free animals have been raised for many years, and does that suggest that there's no sharing of bacteria? Because it would be very difficult, I would imagine.
Well, I think contamination in this field is a really serious concern. For example, I think you're referring, Alan, to the Rob Knight paper in Nature that used the TCGA and looked across 27, a lot of cancers, including things like glioblastoma, and indicated that all these tumors more or less had a microbiome. And there was also the Straussman paper in Science that didn't do quite as many tumors, but did a whole series, and that was done from FFPE and some frozen tissues from multiple different hospitals, as I recall. And again, published a relative amount of a microbiome in each of these different tumors. And what I can tell you is we decided to take the lung data from both of those papers and see they use different methods or different sources and see if we got the same answer at some level. And I can tell you they do not agree. And when you actually pull, say from the Rob Knight paper, the list of bacteria that are listed, at least 50%. We're going through that now and plan to publish this, but many are contaminants, not all. So I'm trying to remember, we made this, it's actually a high school student, believe it or not, at Bronx Science who contacted us for a project during the pandemic. This incredibly smart young woman, Gina Sau, who actually, I think it was last week, presented in the final round of the New York State science things and had applied to the Regeneron competition. But she is actually one who headed up this project with, some help from us. But it's really fascinating how little agreement there is. They agree at the phylum level, which is not saying much, I will tell you. And then from there on down, it dropped dramatically to about under 50% agreement between the two papers. And it's, there's long lists of supposed microbes just for the lung portion. So we're going through them one by one in a systematic fashion, just to make sure we're not misjudging anything. But the ones that I've done so far, it's environmental. And as an ID doc, like, oh my God, I've never heard of these, ones. Are they really in the lung? And I just never know. So I'm concerned about this. And I think we're probably not careful enough, or we're assuming these big data are correct. And I think it needs a much more nuanced eye in order to figure out what's really right if we're going to use this information to try to impact patient care or to understand the pathophysiology. And if I was going to add one more thing, if you'll allow me, I think our biggest challenge is not doing enough human research. I think mouse research is great, and obviously I do a ton of it, but it's complementary. And I think our issue in the field is really doing the very hard work to figure out how to use the microbiome to help patients. I don't know, Noel, if you agree. I mean, you're doing mostly human work.
Yeah.
And I really applaud you for that in trying to figure out these linkages to try to make predictions, try to change, can we change the microbiome? I mean, we're probably going to have our first microbiome therapeutic approved this year. That was in the New England Journal just a couple of weeks ago.
What company is doing the therapeutic?
It's a series 109. So it was a Clostridium difficile product, but really outstanding outcome. And I know that it's going to the FDA soon, and it's hard, from my viewpoint, and I don't know all the data obviously, but it looks like a good bet and could really completely change our approach to recurrent C. difficile therapy.
Cool. Well, I'll get to just one point that you brought up, Cindy, is that I totally agree. I mean, I do a lot of stuff in humans. The challenge is, just like other epidemiology studies, confounding, I think, is a big challenge. And I get a little upset when basic science people throw out the idea that we can not ask questions that observational epidemiology, or like that the answers that we get from observational epidemiology are too biased, then we shouldn't even use those approaches. Like Peter Attia does this podcast, and I totally disagree, because otherwise we just can't ask the questions, because we can't do all the experiments in mice because they don't always translate to humans. So we need to get better at doing things in humans. Totally agree. But we also need good tools to assess for example, confounding by diet. If we do an observational study of the gut microbiome, it's a pretty big potential confounder. And if we do a poor job of measuring the confounder, then it's residual confounding by diet or something of that sort.
Well, it reminds me of the paper that I think was also in Nature, suggesting that the quality of the stool, right, how liquid it was, was a huge, had a huge impact on what the microbial outcome was. And that is definitely not a question that's asked systematically or well across most of these studies.
Cindy, if I can ask a question here, how do you differentiate contamination versus endogenous microbiome? Is there a easy, no easy way, but what's the strategy there?
Well, our strategy was to search the literature for association of the organism with humans, or some data that appeared to link it to a human source. And then beyond that, then we went on the web, and we have a series of approaches to see if we can even figure out what it was. And the ones that I chased down that I didn't recognize were frequently soil, water, plant, things like that didn't resonate with being a stable member of the microbiome.
There was an interesting paper recently that regarding this contamination, because people used to think DNA was also M6A modified. So there was a recent paper which showed that the majority of that is actually from contamination, bacterial contamination. So the DNA is not. So there was like a recent science paper, I think. So again, these contaminations creep in, and it's very difficult to differentiate that.
Yeah. One good thing, Hari, is that in the gut, or fecal samples at least, there's so much bacterial DNA that contaminations, it would be such a small component that it's not as worrisome. It's these low biomass samples that Cindy was talking about, where is there a bacteria there or not? And you can't necessarily say it if you haven't had good collection protocols or sequencing protocols.
Yeah, and do the right. I'm sorry.
Yeah, Noel, you also mentioned that interesting idea that if you're just sequencing, you're grinding up some sample and getting everything that's in there, right? And you don't know that maybe some professional phagocytic cells might have wandered in and they've got some bacteria, on board, right? I'm wondering, is there a way to look at that with some of the single cell RNA-seq data? I mean, it seems like on like a t-SNE plot, you could look at the region, the cluster for, macrophages or something, and see if you see bacterial reads specifically accumulating in there. Would that help get around this issue, or in terms of the source of the contamination source?
Sure. I mean, you could try to culture it, like culturomics, one idea, but if it's culturable, then it's real. But that's hard to do from the way we collect like placenta samples, right? I don't know, Cindy, do you know any ways of getting around that?
No, but there's more and more single cell data out there, and I guess it depends what you're looking for. But I'm not aware of anybody having done that. But I wanted to just emphasize what Noel said. It's the low biomass samples. We're safe when we stay in the colon, and when we get out of the colon, then it gets a lot trickier. And that's exactly the issue, I think, that came up with the placenta. I heard you discussing that in I'm not, that's not my field, but my read of it as well is that was probably contamination at the outset, and at a time when it wasn't really appreciated how many controls you needed to do to try to protect yourself against that.
Regarding the culturomics question, the way we used to do that back in the time when I was doing soil microbiology is basically we used, once we do the DNA sequencing and we see where the 16S falls, we try basically to mimic the culture media of the closest neighboring bacteria. And that's the best way to be able to culture those bacteria that you just have the DNA for. It succeeds something like 50% of the cases, especially if the neighboring bacteria are well characterized and there is good culture media for them. But that requires also validation after you culture those bacteria by 16S again to establish that you really isolate the bacteria for which you had the DNA before.
Good point.
With respect to cancer, and I know I've heard Elizabeth talk about this, but kind of broadening it, there are these observations, right, where in utero exposures, for instance, isn't there the famous famine data that restriction, caloric restriction in youth or maybe even in utero can dramatically affect things like cancer rates and I guess other diseases in adulthood, and how much of that is related to changes, say, in microbiome, and then obviously we want to get into, sort of epigenetics. These are all interrelated and all very complicated. What's the sort of thinking along those lines that predisposing?
No, that I think. Well, I want to hear Hari talk a little bit about epigenetics and whether he thinks those can be passed on from one generation to the next. But I do want to say that although there may not be an in utero sharing of bacteria between mom and fetus, there is a PNAS paper from, I think, 2018 that shows quite elegantly that although there is not microbial sharing of bacteria, the metabolites that are produced from the bacteria in the mom can affect the fetus and program immunologically the fetus also for responses postnatally. So the maternal exposures could still affect the fetus, through metabolite pathways, microbial metabolite pathways too. So I can't speak to the Dutch famine studies, whether they've even considered microbiome as a pathway. What I'm interested in, just because it's in my wheelhouse, is the intergenerational association of obesity between mom and baby. We know that some of it may be due to genetics, but a large part's probably due to environment. Some of that just due to the sharing of household environment and food patterns, but maybe also some due to sharing of bacteria between mom and baby. And if there's truly an obesogenic bacteria, you would expect that to also confer its effects on the offspring. So, and then if we could intervene on that, during parturition or before delivery, then maybe we can avoid colonization of the baby with the obesogenic bacteria.
Yeah. So what's the current thinking on that? I remember many years ago, we did a journal club with the students, which was a fascinating paper in mice, where they took obese and non-obese mice and, swapped their feces.
Yeah.
And then they had coprophagic mice, they'd house them. And then I think the lean mouse tended to somehow be dominant over the obese phenotype. And then they ended up transplanting fecal bacteria from human, I think, twin studies versus, obese versus non-obese. And that would incur obesity into the mice if it came from the obese twin and so forth. And I mean, it was really striking, but that was many years ago. Now that I think about it, I don't remember hearing much about it. Was that
was the Redura paper
going on with that?
Yeah, that was an excellent paper, but it was all diet dependent, right?
Yeah. It was pretty striking, as I recall.
Oh, it was
amazing. You take
Cindy,
bacteria from obese humans and make the mice obese just by feeding them or transplanting the microbiome.
But Cindy, those results have been pretty consistent and then also extended to other cardiovascular outcomes also, not just obesity, and also malnutrition like kwashiorkor. So there's quite a few conditions that were transferable by the microbiome, which
absolutely.
So what about cancer? Is anybody
colorectal cancer
progression or incidence?
Cindy, I'm going to share my screen quick and see if you have any thoughts on this paper, because it's Cindy runs a germ-free mouse lab here at Hopkins. So let's see.
While you are sharing regarding your point on the metabolites, if addicted mothers can pass addiction predisposition to kids, that's definitely point to the case of metabolites being affecting the fetuses as well.
Oh, this is
a 2020
20 paper.
Biomedicine, which is a Lancet open access journal. So they I don't think this is a true germ-free, but they pre-treated with antibiotics and then gavaged with colorectal cancer patients versus controls, and found not only this changes in intestinal adenoma formation, but also gut permeability, production of short chain fatty acids, pathogenic potential pathobionts, and low-grade inflammation, and then something Hari could speak about, the WNT pathway activation. So I thought that this is pretty interesting, but I haven't got into rigorous look at the mechanism or the methods of the paper.
Yeah, I don't know this particular paper, but Joe Zacular years ago in Pat Schloss's lab at University of Michigan had taken human stools, as I recall, from individuals either with colon cancer or polyps. And the outcome was variable. Some did appear to be pro-tumorigenic and some did not. And as you may know, we've taken the mucosa from tumors or mucosa with biofilms versus no biofilms. And in general, those initially, when we grouped samples, those samples that were from humans with biofilms, so these clusters of bacteria that are invading the mucus on the surface of the colon, did induce tumors. Now that was combining patients in early work. When we broke that down, there's no question that some induce tumors and others are either have very modest tumorigenic effects or no tumors are induced. You actually don't have to have a biofilm. We have a biofilm negative sample that is wildly tumorigenic in mice. And we think we understand what may be driving that. Paper we're working on publishing now. But this is very likely correct. I just don't know how consistent it is from person to person.
Now, these are fascinating.
And then the other point I think is translating that to humans. And I think that we've kind of learned that probably fecal transplants can't be extended beyond C. diff for many multifactorial outcomes, too many risks associated with it. So probably the need is going to be to have better characterization of the taxa and then making some kind of cocktail that, seems to be what they did with the C. diff and the series therapeutics, right?
Right.
That will probably be what we'll need also for, I guess, to move the field into humans for obesity, maybe for colorectal cancer too.
I would completely agree. I think FMT can be life-saving at times in C. difficile disease, but it is too crude, not quality controlled. We don't understand it, and it has killed patients. Now we have multiple FDA alerts out about FMT, and the requirements for screening have just grown. So we need quality controlled products, no surprise, to really promote health. Can I ask a question? I am curious. I mean, in the colon cancer data we got from the UK, their very large database that we analyzed, one of the strongest associations with colon cancer was actually BMI. So obesity is, and I don't know the relative, Elizabeth's going to set me straight, but it seems like it's a really strong risk factor. And my question is, do we have some idea of the duration of obesity that then is linked, because obesity is certainly linked to other cancers. How long do you have to be obese? If you lose weight, does it reverse those types of questions?
So great question, Cindy. So big cohort studies that have repeated measures of BMI or other features of body size, those cohorts tend to start in middle age. So we don't have earlier life, which I think is probably quite important. Certainly across adulthood, gaining weight is associated with an increased risk of colon cancer, right? So weight gain, higher weight versus lower weight, and even weight that we would consider to be in the normal range when compared to not underweight, but the low end of the normal range, even that comparison is associated with an increased risk of colon cancer, especially distal.
That's very interesting. Yeah.
Now, does
Yeah.
weight
Does it
gain
matter how
affect?
you lose the weight? Sorry, do you know if it,
Yeah.
because that's the thing that I'm trying to also do with my work is focus on what is the translation to intervention so we could test these things. And does it matter whether it's behavioral weight loss or semaglutide? Like, is that, you think, is it just losing the weight or is it how you lose the weight too?
You know, it's a good question. I do not know, right? So there are some bariatric surgery cohorts out there. I don't know if there's sufficient data yet. That's different than exactly what you asked. That's sort of rapid weight loss. There are multiple mechanisms by which obesity can cause colon cancer, right? And so the question is, which ones are being addressed by weight loss, by what techniques? I don't know. I don't know. And what does weight cycling do? Is weight cycling worse? Or is it better to have transient moments of decline, even if it's, you know, offset by increases again? Don't know.
Noel, so this is a great epigenetic concept there, right? Because the reversal, how long after weight loss can you, what's the risk later on, right? So I don't know about human studies, but in my studies, they have shown that obesity, high-fat diet alters the epigenome and gene regulation of specifically cancer-related pathways and so on. And it stays on, I mean, that epigenetic stays continues for some time, even after reversal to a normal diet. So from a high-fat diet to a normal diet. So there could be a long-term effect of a high-fat diet or high-fat diet induced obesity.
How much might that be linked to obesity or diet related changes in the colon or the gut microbiome and or the metabolites produced by the microbiome? I'm always fascinated by some of the metabolites that you've discussed, and I know there's a lot of focus on things like butyrate, and I mean that's a classical clinical agent in cancer, right, in terms of histone deacetylase inhibition. So obviously it's going to have an impact, I guess naively, potentially on the epigenome of the nearby colon cells.
Yeah,
so that's just too simplistic an idea.
Yeah. Noel, can you move to the I think the butyrate slide? So actually that is one of the key factors that could be playing a role from a diet and so on. And I think this is a study where they had fed mice with high fiber diet, and they see a reduction in colon cancer, actually incidence of colon cancer in the mouse model due to a high fiber diet, which gets converted to butyrate. So butyrate is one of the key factors, but there are a lot of other metabolites which have been known to directly interfere with epigenetic machinery. There's propionate, there is crotonylation, and so on. So many other factors actually have an impact on the epigenome. So diet plus the good microbiome can actually also reduce colon cancer risk, reduce the colon cancer growth, and actually increase response to drugs, chemotherapies, and so on. So one of these pathways is through these short chain fatty acids.
And is the microbiome or changes or changes in the metabolites, is this also affecting local inflammatory processes, which, you know, inflammation and cancer risk is somewhat linked as well?
Yeah, so these components, they have impact directly on not only on the epigenome. So that is where it gets complicated, not only on the epigenome, but then it also it has immunomodulatory roles. Some of them are pro-inflammatory, some of these metabolites are anti-inflammatory. So that actually has an additional twist where it can actually alter the immune profile, inflammation, cytokine signaling, and that can actually either have a pro-tumorigenic or an anti-tumorigenic effect. And butyrate is one of these metabolites that has actually has a pro-inflammatory effect, but is actually protective for works against cancer cells.
I think there's a little there is a paper that also suggested it's a 2021 paper, Ari, and I don't think I shared this with you yet, but there's a little bit of inconsistency in the literature with butyrate and colorectal cancer risk. And part of this might be also due to where the butyrate's measured, especially with human studies, because if it's measured in stool samples, that means it's being excreted. If it's measured in circulation, that means it's been absorbed, but it's post-utilization by the colonocytes and post-utilization by hepatocytes. So it's influenced by metabolic pathways already. So it's a little bit difficult to know exactly what stools or what biospecimen is the best way to measure it in human studies. But I completely agree. Butyrate's one of the key kind of metabolites, I think, that's thought to mediate the microbiome to colon cancer for sure.
So basic studies not been done yet, trying to correlate stool butyrate with, say, systemic circulating butyrate. I mean, naively, I'd expect if there's a lot in the stool, then it's going to be absorbed, but maybe the biology of the colonocyte.
There's been some studies mostly looking at like acetate, but the acetate's a little bit more readily absorbed and circulated. And I think the more excretion there is, the less there is in circulation from just direct administration of acetate, which would suggest that measuring the stool might actually be measuring secretion. So it could depend on how much somebody sheds versus their, there's different ways that it's being excreted. Like in a human, you might be excreting a lot of absolute acetate or butyrate because you're consuming more calories, because you're consuming more fiber, or because you're not absorbing it because you have some other issue with absorption of and utilization of the short chain fatty acids.
As far as I recall, for these short chain fatty acids, both butyrate and propionate are consumed within the gut, and there is little go into the circulation, unlike acetate, which is circulating more. So I don't know if that correlation between the stool and the circulation butyrate will lead to any tangible data. That's an open question.
Yeah, that's right. Butyrate, I think, is like 90% absorbed and used by colonocytes. But there is still, I think 5 to 10% that makes it into circulation. And there's, already some work showing that there's receptors for butyrate throughout the periphery, including in skeletal muscle and adipose tissue, and even I think nephrons. Jennifer Plesnick, who's done some work on finding receptors for short chain fatty acids in nephrons, suggesting that might be a pathway to high and low blood pressure. So I think that yes, it's definitely known that acetate is more readily circulated, absorbed and circulated than butyrate, but butyrate still does make it into circulation. So I don't think we could rule out that still has a potential effect.
And also, it's kind of interesting because butyrate, there's mice studies which has shown that depending on the genotype of the mutations, butyrate can have a pro-tumorigenic or an anti-tumorigenic effect. So there was one study which again, high profile, I think it was a cancer discovery, where they had shown that butyrate in an APC min mice with MSH2 mutations, the DNA repair mutations, that actually, in that case, butyrate has an tumorigenic effect. So which goes in contrary to many other studies. So the key conclusion there was it depends on what kind of genotype these cancer initiating cells might have. And I think that might somehow build into also the immune status of the the immune microenvironment there.
So maybe Cindy would comment on this, but it's my understanding that as a model, so the APC min mouse, the APC is a critical, loss of APC is critical to human colon cancer. But in the mouse model, I think these mice tend to get small intestinal cancer, not large intestine, right?
Yes.
So how does that impact where the bacteria are, where the butyrate is, and what's going on with that in terms of the microbiome? And it's pretty, seems confusing.
It is very confusing. And it is impressive to me how useful the min mouse was before we knew how to convert it into a colon tumor model.
Oh, how do you do that?
Noel, can you move to one of the slides? Cindy, I had your slide there, so I thought that'll be useful for you. Yep, there we go.
I didn't even know the slides were being shared, so I feel really stupid. I just found out I have the second screen.
Yeah, it's almost as if we set you up to ask.
Very
these questions,
helpful.
Alan. Perfect.
Noel, the next
Thank you.
slide. Noel, the next slide, I think.
Next one. All right.
Yep, there you go.
Yeah, so, if we use various bacteria, and as I mentioned, even human colon mucosa with biofilms, whether it's from normal mucosa or from cancers, or the normal mucosa from individuals with cancers, we can drive colon tumors in the min mouse. Now, the issue is, are any of these bacteria actually initiators, right? And that is as I've learned the hard way, a very, very difficult question. The only one there's only two, to my knowledge, that have been really studied in enough depth to even begin to try to sort that out. And one is the PKS positive E. coli that make this DNA damaging toxin called Colibactin. And that particular bacterium in a highly artificial system was, able to induce a specific mutation in DNA, but the system used was done by Hans Clover's lab, was really highly artificial. It was a collonoid. They infected it, they uninfected it, they infected it. They did that about 8 times in 6 months. And then they found this mutation. Now, the upside of that is then they looked at very large sequencing studies and have found that mutation in human colon cancers. And the fascinating thing is, for me, is that it seems to track to the APC gene. So if you find it, it tends to be, I forget the exact numbers, but tends to be in the APC gene, which I think raises really interesting questions, because obviously both in somatic and hereditary colon cancer, the APC gene is really important. And if you look at FAP, there's like 700 different APC mutations that can be associated with that hereditary syndrome. Now, what we don't know is if you're walking around, and PKS E. coli, it turns out, are incredibly common in the gut of humans. And we don't know a lot about these strains. So we decided we would look for these in our colonoscopy project, which was a prospective biopsy-based. We got stool from about 70% of the patients. And when we thoroughly examined 100 people using both culture and molecular means, we found 43%, or 43 patients were carrying a PKS positive E. coli. And now they're not.
And it wasn't a contaminant?
It's not a contaminant, because we grew about 23, 25 of them. And now we're sequencing them all, and we're beginning to test their virulence capabilities. But what we don't know is, does that associate with polyp formation over time? So we have about 60 individuals where we have sequential colonoscopies now, and we're going to, and some of them have the PKS E. coli. So we're sequencing the polyps to see from people who are PKS positive and PKS negative to see if the mutation will track with the bug. We have no way to measure serologically exposure, previous exposure to PKS E. coli, because the colibactin is made so fleetingly that it's not immunogenic as far as we know. And no one yet has, it takes about 12 or 13 genes to make the molecule, and no one has succeeded in purifying it yet. So there's a paucity of tools. There is an association of one gene contained by these bugs called the FIMH gene that regulates adherence. And there has been an association between the degree of adherence and the likelihood of someone getting this ileal Crohn's disease, which actually is how the organism was first recognized, recognized in a subset of Crohn's disease, which of course is a precursor potentially to colon tumors. So I got totally off track. I apologize. If you ask me about the min mouse, as is, those small bowel tumors are actually pathophysiologically not very helpful. We've studied them. They have a completely different immune makeup than the colon tumors. So it's very impressive that it was so useful over time, even though it's really way off the track of what we see in the colon. Sorry about that. I'm very enthusiastic about some of these bugs.
We're learning a lot here. This is great.
Did you want to talk about this slide at all more?
I think, Cindy probably covered, basically Cindy covered it, right? So in this model, what they have shown is that when you introduce this ETBF, you have distal tumors in the colon. So compared to the APC min mice in general. And what is interesting here is actually when you introduce a BRAF mutation in this model, the tumors move up more towards proximal side. And actually that is interesting because in human cancers, majority of the BRAF induced tumors are in the proximal colon. And that is kind of very interesting that just by this combination of bacteria, APC min background, and introduction of BRAF, it moves, the tumors move towards proximal side. Actually, if you move to the next slide, and that is kind of, and we think that there is probably a strong epigenetic basis to this too, because we had shown that in using organoid models generated from proximal colon, that you need a set of epigenetic alterations required for these colon-derived stem cells to undergo tumorigenesis in response to BRAF mutant activation. And I had a list of notes on the right, but maybe if you click, does it show up anything? There you go. Yeah. So actually, what's kind of currently what we are learning is that we have been comparing proximal and distal colon-derived stem cells or organoids, and what we see is that that's actually probably a pre-existing epigenetic landscape of the stem cells from proximal and distal colon, which has a complete different response to BRAF. So when we, in the proximal colon, what we notice is that if you inactivate one of these genes, developmental regulator CDX2, it can immediately undergo tumorigenesis in response to BRAF, but the distal colon does not. And this gene is downregulated in the proximal colon even in human cancers. So there is some relationship that there is this important transcription factor, which is CDX2, and proximal colon stem cells are more sensitive to downregulation of this transcription factor. And I've always kind of wondered what happens to this in the ETBF model, Cindy, because it might be interesting to see if ETBF has a direct impact on the transcription factor regulatory networks, and that is how it kind of shifts from a more distal to a proximal colon phenotype. So yeah, this question keeps coming up whenever we think about it, and whenever I present the data. Is the microbiome involved in actually also altering the transcription factor repertoire?
Yeah, we're interested in pursuing this model because not only does the site of tumor change, but the immune response changes, the histopathology changes, and now the tumors are sensitive to checkpoint inhibitors, to anti-PD-L1.
Yep.
But and ETBF is very much changes the epigenome. So Harry, we should definitely talk about that because you could probably really help us sharpen some of what we're doing, trying to understand what happens here. I consider this good proof that single organisms in the microbiome can dramatically interface with the oncogene makeup of a tumor, and that can in part dictate the biology. Now, admittedly, it's a mouse. It's going to be much harder to show that in a human. But conceptually, I think it raises that possibility.
With respect to the mouse model here for epigenetics, I know you've got bringing up this issue of changes in DNA methylation status with aging in humans. Does this recapitulated in the mouse? Is it like one of these things that happens, on an accelerated sort of proportional time scale to the mouse lifespan that you'd see it in the human lifespan? Or do they not undergo these same types of epigenetic changes with mouse aging that we see in humans?
That's very kind of a very fascinating question because the mouse they also undergo the same sort of epigenetic changes in similar regions during aging as do humans. And they undergo these changes at a much faster rate than human because they according to proportional to their lifetime. If you really calculate the rate of changes, it's much faster in mouse.
So their epigenetic methylation clock runs. Have people done that in the mouse, like the Horvath and that type of thing? Is it?
Yeah.
Like so the slope is totally different in a mice, but
the slope is completely different. And if you adjust for lifespan, the slope remains the same, which kind of tells you that there is some sort of clock that is adjusted for lifespan here. And I kind of, again, it comes to metabolome, metabolite, so on, because I think it's probably linked to the metabolic rate because the rate of metabolism is very different, which much faster in mouse compared to human. So here is like a thought experiment that I've always think is, can we grow in some sort of humanized mice model, human cells, and for a period of at least 6 months to 1 year, and then kind of see what is the rate of the methylation clock, how it works in a human cell growing in a mouse.
That's a great idea. I love it.
And that will really actually nail it, whether how the metabolic rate has and the genome and the host genome has an impact on this. So yeah,
it would have to be a normal tissue. You can't.
Yeah, it has
go
to be
6 months with a cancer. The tumor is going to kill the mouse, but
it has to be a normal.
Can you do that, Cindy? Can you do that? Can you transplant normal human tissues? I guess probably the old studies were probably skin grafts, right? Across species barriers, if it's immunocompromised, which introduces a bit of
usually use nude mice
complication. But
right?
Can you put a piece of human kidney in the mouse and let it just live there for like 6 months and then pull it back out?
I think you can in a nude mouse, can't you? That's what I forget what they call them. The human. They put cancers in.
Actually, for prostate, I do know they've done this, right? So prostate, this guy did these studies years ago. They could take, I think they would take human prostate tissue. You stick it under the kidney capsule, and it'll grow and it'll stay. And that wasn't tumor tissue. That was, gosh, was it coming from humans though? I can't remember.
Isn't that like xenograft?
Yeah, but most xenografts are cancer, right? Does anybody xenograft normal human? Well, what about the humanized mice? When you reestablish the hematopoietic system, is that a one-shot deal, or do they just completely ablate the hematopoietic stem cells and replace them with human hematopoietic stem cells?
Yeah,
the transplant people must know this. Yeah, right.
Yeah, you replace with human hematopoietic.
I think this is a really cool experiment, Hari. You should do it. You got to figure out how to do this.
Yeah, I've wondered whether we have thought about the humanized mice, whether in the humanized mice, because now it is replaced with the human hematopoietic stem cells, but I'm not sure how long they last. So that is something which, yeah, I want to talk to some experts around here and see in the center how long do they last, because if they last for 6 months and 1 year, that'll be a neat experiment to try there.
Yeah, that'd be fascinating. Maybe you should explain. Maybe we might have gotten a little bit ahead of people talking about these methylation age clocks. Can you briefly describe what these things are so everybody knows what we're talking about?
Yeah, so let me check if I had a slide here. Give me a second. So yeah, so
Do you have one earlier?
I had one earlier. Let me switch to that slide. Okay, so here, Noel, can I share my screen?
Yep. I was also going to say that microbiome also goes through a life cycle. So we could also do the same mouse experiment, human versus mouse, to see whether the same life cycle of the microbiome changes in the mouse the same way.
Oh, cool.
Noel, sorry, can you?
I'm trying. 1 sec.
No, I don't think I put that slide. So if we can put, if you stop screen share, I'll just share my screen.
Okay. Trying to stop screen sharing right now. For some reason, I'm not.
So I can just keep talking. And so basically, I mean, there is this methylation changes, even chromatin changes that is known to occur during aging, and and it is universally it is observed that there are changes in. There you go. So this is again a cartoon slide, but basically,
They're the best.
Say it again.
They're the best. The cartoon.
Yeah,
slides.
they are the best. Because it's a very well-known fact. So what happens is there are regions in the genome, like the repetitive elements, the gene bodies, intergenic regions, and so on, which lose DNA methylation. And generally, the promoter elements which have high density of CpG residues, they gain methylation. So this is generally observed across tissues in normal cells during aging. And there are various molecular mechanisms underlying it, which we can actually get into the details of how that might be happening. But what is interesting is actually this also occurs in cancer. Cancers have an exaggerated phenotype of such molecular changes that you observe in aging. So based on these normal methylation changes during aging, the flux that happens at loss of methylation and gain of methylation, there is this, people have derived this epigenetic clock, which actually very accurately within 2 to 3 years error, they can predict the age of an individual just by getting blood or specific tissues from the body. So you can actually predict the age just based on DNA methylation. And what is similar, such clocks actually work also in mice. People have derived mice DNA methylation based clocks, and that's what kind of shows that the mice aging occurs. Mice methylation rate is much faster than human methylation rate.
So if you plotted the mouse data on using the lifespan of the mouse proportionally here, that shape of that curve would sort of look very similar then.
It'll look very similar. I'll pull.
So the rate's got to be a lot faster. Yeah, right.
Yeah, it's not in this slide deck, but let me try to pull that, and I'll just kind of.
Sorry, I'm curious about how modifiable this notion of epigenetic clock is. Like, is this something that is a natural phenomenon or something that could be prevented if we were to optimize lifestyle? Or
yes, so that's where it becomes interesting, because you can actually, just by modifying diet, a caloric restricted diet reduces this rate of methylation change, and actually it can switch back your age, the methylation age. So the term that is generally used for this is methylation age. And the lower your methylation age compared to your chronological age, you are more healthier. And it can be modified by a diet, a caloric restriction diet, restricted diet reduces your methylation age.
mm-hmm.
I don't know the details, but due to the caloric restricted diet, there's also alterations in the microbiome. We don't know exactly what the the components of the microbiome are, but as we move towards a healthier diet, the microbiome population also changes, and actually that has been associated with reduced age.
It's interesting because there's also microbial age that is correlated with age, but there's a lot of heterogeneity in it. And we've started to look a little bit at microbiome age in infants, and our hypothesis is that earlier maturation of the microbiome is not a good thing. It might suggest that they're being introduced to solid foods too soon or other things, and then that would be associated with later life, diseases. But there's also like the mismatch between chronological age and microbial age might mean something also similar to epigenetics. So I think
microbial age there, does that mean the composition of the different species present, I assume?
Yeah,
yeah,
exactly.
How about things like breastfeeding versus formula feeding? Does that change it?
Formula feeding and introduction to solid foods advances microbial age faster than if somebody's breastfed. Wow. Breastfed tends to drive proliferation of bifidobacteria and less diversity, probably to protect the gut microbiome from pathogenic species. So those are some of the factors that have been identified with microbial age. But I also think it's, when we're thinking about in trials, what we can look at as outcomes that might be meaningful biomarkers, like can we modulate epigenetic age or microbiome age, and are those meaningful enough surrogate markers for long-term outcomes?
I have a question, if that's okay, that might, I started thinking about this when you mentioned the cesarean delivery versus vaginal birth. And that might have something to do with some of these epigenetic clock type experiments that we've been discussing. Has anyone looked at, or is there any, if you did a thought experiment, right, and you looked at an infant that was delivered by cesarean section versus one that was delivered by vaginal birth? In humans, obviously, you might not do this right away, but in mice, maybe you could do this, where you would expose the litter to bacteria at different times, so either at birth or later. Does it make a difference when you would expose those litter mates to bacteria? In other words, is there something that gets fixed, perhaps epigenetically or genetically, in time? And because if that's true, if there's some way to find out when the exposure might matter, if it does, then that might mean you could intervene at some age. The pediatrician might be able to intervene. Does that make sense?
I think that, to me, I'm interpreting that as does sequence of colonization matter, and like also the timing of initial colonization, does that matter? I think that's a great idea, because there's this notion that it depends on the order in which you're colonized by bacteria. That might matter also, but it's hard to test that, I think. But your idea, to do that in mice could start to get at that question, I think.
Yeah, and it also might point to the biology as well, as to whether there are points of no return, whether something is fixed or not. Mm-hmm. And if you could intervene later, that might tell you about the biology as well. So, yeah, thanks.
Yeah, there's a paper from Laurie Cox and Marty Blazer, 2014 Cell, which suggests that, so they looked at a control group of mice, and then they looked at a group that was exposed to penicillin for 4 weeks, 8 weeks, and 28 weeks, I think, and then looked at development of the microbiome, and then also development of fat mass and non-fat mass, and found that those that were exposed to 4, 8, or 28 weeks of penicillin compared to control all had initial changes in the microbiome, which you'd expect. But then they also had additional fat mass accumulation, suggesting that antibiotics through the microbiome might play a role in fat mass accumulation. But then when they looked at the mice later on, past 28 weeks, they found convergence of the microbiome, but the differences in fat mass and adiposity persisted after that, suggesting that like there's this early life window that if you disrupt microbiome development, it has a legacy effect on metabolic outcomes that even when you cross-sectionally would look later in life, you may not be able to pick up that there were early life differences in the microbiome, but there are phenotypic differences. So, and then your question is like, does that persist? And then also, I think that the extension is there an epigenetic mark or imprint that you could pick up later that is due to the change in the microbiome early in life?
Yeah, fascinating. Yeah, absolutely.
I wonder if you could also use like FMT experiments and look at engraftment from fecal microbiome transplants to see whether, if you try to introduce a whole new microbiome and engraft it into an existing community, how well does it settle into the community? Certainly in a really damaged community, like somebody with recurrent Clostridium difficile, you can get that new community in. But I think trials in like Crohn's disease and in IBD have failed because they couldn't get engraftment. And so that might be a human way to try to answer that question.
Yeah, definitely. That's a good point, Justine.
Noel, do you want to introduce Justine?
Yeah, absolutely. I was going to say, so Justine Debelius is a new hire in the Department of Epidemiology. She's got her PhD with Rob Knight, did a postdoc with Rob Knight, and then expanded her horizons to go to the Karolinska Institute and get training in epidemiology. So she's addressing a lot of those kind of limitations in microbiome studies, or the microbiome trying to do epidemiology, addressing it from both angles, I would say. And so we were, fortunate to hire her, and she just started in February. So she can definitely come on board for people who are looking for more bioinformatic, biostatistical help in microbiome studies. Justine, did you want to say anything else? I don't want to put you on the spot.
No, thank you for the really kind introduction.
Welcome back to Baltimore too. I loved your faculty profile. Justine's originally from Baltimore. Now she's back.
Great. Welcome back. People always return, strangely enough.
Is that the background you have now? Is that Baltimore or?
Oh no, that I just needed a picture. That's Stockholm. That's like a castle.
Nice. Okay. That's what I was wondering. Where is that in Baltimore?
Yeah, we're flooded. You didn't see all the rain we had today, Hari.
That's No, I thought she also had a picture, but I was wondering where it is from. Yeah.
So I wonder in your description of lack of colonization, what is that because of competition, or why wouldn't they be able to clear the gut first out with antibiotics or something? And why would a new or microbiome fail to colonize?
I think that's my interpretation of the literature. So there's been better engraftment of FMTs, fecal microbiome transplants, in the C. diff experiments because they've already gone through so many doses of antibiotics for C. diff. Whereas when you go into a model like Crohn's disease or IBD, and they've had less pre-antibiotic exposure, there's less engraftment, and it just seems to be less successful. So the thought
they're just not tolerant of antibiotic treatment. You can't hit them up front, or is it because it's such a highly inflamed environment that
it's because they're not pre-treating with antibiotics in those diseases, because of probably they don't want to proliferate antimicrobial resistant genes. It's hard to justify scaling pre-treatment with antibiotics in light of AMR. I think.
Yeah. Okay. That makes sense. Yeah.
Same thing with, I think, probiotics. Maybe could even work better with a pre-treatment with antibiotics, but then it's like, is that something that we want to really scale? Probably not.
Yeah.
And there's not good evidence for probiotic engraftment that I can find. Like if you're going to take a probiotic, you just have to continue taking it. And whether the probiotics you add in adults are even the appropriate thing. Like I don't drink enough milk to justify taking bifidobacteria every day, but CVS definitely wants to convince me I should.
Yeah. Yep. And there's some, Justine, I'm not sure if you're aware, there's some evidence that if you look at who does better with engraftment of certain species, it's people who have already developed or have a niche for that bacteria. Like it could be at small relative abundance, but then that shows their capacity for being able to tolerate the bacteria. So they actually have better, I think, engraftment of bacteria. But I think that's only like one study that showed that too.
Cool.
So, I was kind of thinking about this discussion and the antibiotic treatment. So is maybe Liz or Cindy might know, are there any data sets that we can look into where early life antibiotic treatment can be related to cancer risk, colon cancer risk, but especially the early onset colon cancer?
So really quick, I just noticed in the chat, Cindy had to leave.
Oh,
She said, my apologies. I have to go to an event at 5:30, but I
Yeah.
want to make sure I got this in. Like, she said, thank you all, especially Noel and Hari, for this very interesting discussion. So unfortunately, she's not
okay.
here anymore. I won't say she's not with us anymore. That has connotations, but
Yeah.
but I want to make sure you guys knew she was very appreciative and so it's up to you, Elizabeth.
Yeah. So, Cindy and her team have used the what's it called? It's not the UK Biobank. It's the general medical, where they have all the medical records. So they've done that to look at antibiotic use and colon cancer, but I don't know that they specifically looked at early life use. Early life use is probably episodic, right? A kid has some kind of, ear infection, whatever. So I don't know if there's enough like duration of use. That would be an important question, right? Is one dose sufficient? When I say dose, I mean course sufficient. I mean, I guess the research that I'm aware of shows that you can transiently change the microbiome even with a single dose, right? Not dose, but course of antibiotics. I don't know, like in childhood, like what happens? Does anybody know?
Yeah, so there's a transient effect with a single 1 week course of antibiotics in childhood. But then I think the thought is that most of your microbiome is restored post-treatment, especially if you have a pretty robust immune system or microbiome at that point. But then the thought is that it's like subsequent doses of antibiotics will have cumulative effects and maybe even have a synergistic effect where you get to a certain point where there's certain bacteria that will never come back. And there's also been, in terms of phenotypic outcomes, there's been like a dose response study looking at number of courses of antibiotics in the first 2 years with greater BMI Z-score in kids, suggesting that like the more courses, it may take 2, 3, or 4 courses to really have an effect on additional BMI Z-score. But I think that those types of questions, looking at long-term effects and whether there's an imprinting on the epigenetics, and then also some kind of legacy effect that would lead to later in life colon cancer risk, I think that would be really interesting.
There is also the scarring in Justin Sonnenberg's mice, where he did a generational experiment, and I don't know if he did. I think he did 5 generations, but I might be off on that, and fed them either high fat or high fiber, low fiber diet. And you could see the cumulative loss in these mouse generations over time based on treatment and diet. And so there's likely an interaction there as well, that more resilient people are going to have a more supportive diet, and you might be able to fix the effect of early antibiotics by feeding kids correctly.
I'm glad you brought that study up. That's a really nice study that they basically were also testing whether you could restore the depleted microbiome, and your ability to restore it through a healthy diet depleted or like was reduced with each subsequent generation. So first of all, it's like there's probably evidence there that there's vertical transmission of the loss of microbes, and that as you get to generation 5, you can no longer restore the original microbiome through diet alone. So
Actually, we just published a paper a few months ago about the use of antibiotics in sickle cell patients and the microbiome, although it's not in the context of cancer, but at least in sickle cell patients who take a lot of antibiotics early on, what we are showing is that their gut microbiome seems to be stuck at early age microbiome. Now, and of course, this is further compounded by the ischemic and hypoxic conditions in sickle cell patients. But how would that relate to cancer risk? That's an open question.
I'm a little confused by this generational mouse study. I mean, did they take like a late generation litter and, you know, stick it in with a not late generation mother for weaning or something and see what effect that had, to see if it's the mother, or is it something that's been imprinted on the neonate itself?
I think that from my recollection,
What's the mechanism? I don't understand the mechanism.
They treat it, so let's say that generation 1, G1, they treat the mother, they give birth to a litter.
Okay.
Then that generation gives birth to another litter. And they do this for 5 generations.
And they're treating each generation?
I think they do treat each generation. Yeah, I think they each, but it's more than an additive effect of each generation, a loss of microbial diversity. But is that right, Justine, that they treat each generation? And then there's evidence that
they switch, like they switch generations. So generation 1 and 2 might be treated, and then generations 3, 4, 5 wouldn't be, to see the recovery. But in terms of inheritance, there's also that study that just came out of, I think the race lab, but it might be Gwen Filoni's own group in Belgium, looking at, like, I think they had 3 or 4 generations of inheritance, and they could see the inheritance in adult microbiomes, but not like 3-year-olds and younger. So there has to be some vertical transmission effect somewhere in there.
I mean, presumably that's epigenetic, right?
Well, you can have direct microbial transmission, mother to child, certainly. Like, I'm trying to think who did this experiment, but living in the same household, you share microbes.
Yeah, that's true, I guess. Yeah,
we have some data that's unpublished, Justine, that it's kind of like, it's a sample of a birth cohort 5 years after birth, mother-child pairs, and we're just like, oh well, we're bringing them in for a visit, we might as well collect stool. So we have about like 200 mother-child pairs, and we've kind of just looked at like Bray-Curtis distance, which is a measure of beta diversity, and clearly mother-child pairs are way more similar than mother-mothers, who you might expect like those are 2 adults, or mothers and other children, or children and other children. So not genetically related. We can't tease apart whether that's due to genetics or just living in the same household, but there's a much higher concordance of bacteria and phylogenetic similarity between mother-child pairs if they're related.
Yeah, it might be worth looking for like either twins in that study or twin data.
What about adopted? If you had an adopted child versus the maternal's own child, I feel like that would be the perfect comparison. If you can get them adopted early enough and age match somehow. I guess it's getting harder and harder to do. All your epi studies are tough.
But then also, if we wanted to like think about this with colorectal cancer, you know, there's a genetic component, but is there also evidence that maybe vertical transmission of bacteria may play a role? Be a hard study, but maybe the Dutch might have the data. Who knows?
I think there you need twin pairs, or the Swedes. If that data, they have a population-based screening cohort with, I think, I don't know, it's a huge cohort, 30,000 fecal samples collected.
Wow. Jeez.
So we'll see how much of that gets totally worked through, but yeah, that might also be a thing to look at when that data gets pulled together.
Maybe we need to invite one of your former colleagues to come and give a talk. I'd love to hear about that large of a sample size.
I'm happy to invite them, but it might need to be earlier in the day.
Yeah, no problem.
Justin and Noel, I had a question about, again, the transgenerational effect. By the fifth or sixth generation, do these mice show any diseases?
I don't think they were looking phenotypically. I think they were just looking at the microbiome and cumulative effects.
It's published, I think, in Nature or Science. Yeah, I don't know if they looked at outcomes either. I don't think they did.
Okay. So Noel, like a thought experiment again, and maybe in the context of your sensors, that's kind of interesting to look at metabolites. Is there a way one can to tease apart the role of direct contact of the microbe with the epithelial cells, or translocation into the bloodstream and so on, versus metabolites? Is there a way we can actually, grow microbes in the colon, let's say, but not let them leak out from some such matrix? So can we look at what is the impact of metabolites versus microbial contact?
Yeah, they have like robo guts, which are probably the closest you can get to that type of model, where you simulate what a human gut is like, and you can introduce any kind of exogenous factor to the system, and then see how that changes composition. You could see how it changes metabolome of the robo gut.
What's a robo gut made out of?
I don't know exactly how it's made.
It's artificial?
It's artificial, yeah.
Oh, that sounds pretty sketchy.
I've never seen one in person. Justine, have you seen one?
My old lab was setting one up just as I was leaving. It's kind of like a big, I don't know, tank, and you can seed it with a fecal sample. So you can start with a donor sample, and then they'll grow that donor sample out and see what happens. But
is there human tissue involved in it? Okay.
Well, to some degree, because you're getting the fecal sample, and so there's like human cells that get sloughed off into your stool. Okay. But
yeah, probably dead, right?
I know, it's like it must be as close to the human as possible. Otherwise, it's just a red flag for anything you put into a paper. I mean, I wonder how much it helps, but I'd like to see one in person before I can criticize.
Curious, yeah.
But you could also, another way to maybe, because are you getting at a bias due to what's excreted versus what's happening in the gut, Hari, with your question? Because another way that you could do it is you could swallow, they have a swallowable thing that collects microbial samples from different parts of the, intestinal tract, small intestine and large intestine.
Yeah. No, what I was kind of thinking was because there is so much epigenetics, probably a direct impact on epigenetics is through metabolites. So to tease that, and the role of let's say colorectal cancer risk, or like progression of colorectal cancer and microbiome that we have been discussing, how much of that is directly by interaction of the microbiome with the epithelial cells and the immune component versus metabolites produced by the microbiome? So is there a way, and we cannot mimic the metabolites by culturing the bacteria outside and somehow gavaging the metabolites inside? It might, that might not be the perfect experiment. But then I was thinking, is there a way, like some matrix or capsules, or where we can grow the microbiome inside the gut, and then actually follow the phenotypic changes, what happens to tumor initiation and things like that in appropriate, of course, in mice models. But yeah.
Not sure. And those types of experiments are a little bit tricky for an epidemiologist to really put too much mind on, I guess. I don't know, because for me, it's like, I'm not sure, like, I feel like a lot of things are probably due to metabolite formation, secondary to, microbial production metabolites, but there's also the examples of like the translocation of bacteria into circulation causing low-grade inflammation.
Exactly. Yeah,
Yeah.
that's right.
You could do like a sodium butyrate feeding experiment, and because that should be a direct epigenetic marker, and certainly a lot of the bacteria of interest are butyrate producers.
Yeah.
And so if you fed your mice, butyrate or a combination of butyrate, propionate, and acetate in whatever ratio, you could look for epigenetic modification. And there's a lot of diseases that are linked to differences in short chain fatty acids.
There is one like low-hanging idea that we have to do. So that's one thing we should try.
So this reminds me of a phenomenon that we haven't discussed yet, which is leaky gut, right? Leakage of the crossing the epithelial barrier. What's going on with that? Is that a real phenomenon? I think they measure it with some type of dextran or something, and you see it, something so big that it shouldn't be able to get out, and it gets out. And what's the consequence of that in terms of cancer, epigenetics, or what's the skinny on leaky gut?
So I think that's, it's something that people are interested in studying. SCD14 is one measure of that. Lipopolysaccharides and lipopolysaccharide binding proteins. We've looked at that in one behavioral study, a behavioral lifestyle intervention study. We actually did find that the behavioral lifestyle intervention, including a DASH diet, which is like a fruit and vegetable forward diet, and weight loss, decreased lipopolysaccharide binding protein, suggesting that it decreased permeability of lipopolysaccharides. So that was interesting, but I think it's still hard to know exactly, where like the mechanism of how it's happening. I think it probably has, Justin Sonnenberg is another example here where they suggest, and I think they've shown in a mouse study, that if you deplete the fiber content of your diet, then the mucin will become the substrate for the bacteria that rely on mucin, right? So the mucin degrading bacteria will break down the mucin or the mucus of the gut, and that would lead to leaky gut. Because if you're not providing it through your dietary exogenous substrate, then it has to come from somewhere. And then over time, that, degradation of the mucin layer is going to lead to leaky gut and translocation of bacteria, or, the, lipopolysaccharides into the circulation. So I think that's like the theory, but mechanistically, I'm not sure how feasible that is.
The other one I think isn't talked about as much, but I'm really excited about, and I don't know if you could explore in mice or epidemiologically, is oral translocation all over the body. Because the bacteria that keep coming up, like if you look at what's on a colorectal tumor, it's Fusobacterium nucleatum, which is an oral bacteria. And then Fusobacterium nucleatum is hanging out with a bunch of oral friends like Negativus and Streptococcus, things like that, that should live in your mouth, but not in your gut. And you see this oral linkage again across diseases. In rheumatoid arthritis, you have an oral translocation. And then when you treat it and fix and decrease the inflammation, you see less oral bacteria and you see a correction. So, I don't know if you could try that in mice, change the oral microbiome and see what it does to fecal, except that you've got, I guess you have coprophagy in your mice.
Yeah.
Which is a delightful feature.
So Justine, I can't wait to talk to you and Noel and others. So we have a whole line of research, this is in the ERIC cohort, looking at the the oral microbiome, but done the old-fashioned way. So looking in subgingival plaque. This is in a long-standing dental exam that was done in the ERIC cohort in relation to distant sites for cancer. So lung cancer, GI, and colon cancer. We've got studies showing associations between periodontal disease and future risk of cancer of those distant sites. So I would love to get involved in conversations about trying to understand, is it the metabolites? Is it just an altered immune response because these people have long-standing periodontal disease? Is it that these bacteria are just translocating somewhere else and causing, aspiration or translocating to the, somewhere else and causing an inflammatory response? We don't have answers. We're doing observational research, right? So again, it's direct measurement. It's looking at antibodies against these agents, and then a clinical dental exam where they have periodontal disease and severity diagnosed.
Elizabeth, can you just give like a 1-minute synopsis on what the ERIC cohort is?
Yeah, so it's a large prospective cohort study. There's 4 sites in the US. It's been ongoing since the late 1980s. It's a cardiovascular disease cohort. We enhanced its infrastructure for cancer research about 10 or so years ago. And so now we're able to leverage everything that's been collected in the cardiovascular, kidney, and diabetes space for cancer research. So it's actually very exciting, right? Because, we in the cancer world, we usually have one measurement. ERIC, for whatever reason, has multiple measurements, everything. They have blood samples at each visit. It's really a remarkable cohort study.
What cancers are you able to look at in that study?
Well, any of the common cancers, so prostate, breast, colon, lung, pancreas.
Very cool. And I know they have a small subset has gut microbiome samples as well, I think.
I don't have a gut. They've got the stuff that Demers has been doing.
Brian
I thought that was, I thought that was that's saliva based.
Katie Meyer's doing work, but maybe that's cardia.
Yeah, it could be. I haven't heard anybody say gut, but my goodness, if they have gut, I would like to know that so that we can relate it back to what's going on in the oral cavity. Yeah. But it was dentists that added the clinical dental exam and the collection of the plaque for evaluation.
Yeah, if only we could, measure that Fusobacterium nucleatum or other oral bacteria, and then look at a dental intervention to see whether it reduces its translocation or engraftment in the colon biofilms.
University of Maryland has a dental school. So John Grootman, remember Norma, when John Grootman said we should go over there and try to collaborate with them, especially for populations that maybe haven't had access to dental care. So the state of Maryland has laws now to make sure that people have access to dental care. So newer people who might have a higher likelihood of having periodontal disease, and therefore some of these agents, he thought we should try to do some kind of intervention study. Our colleagues at the University of North Carolina who started the dental exam in ERIC told us that if you treat one side of the mouth for periodontal disease, the other side will clear. Wow. And he thinks it's because you get a local, you basically cut the gums, right? When you're trying to scrape off all that stuff. So then you get a septicemia, a little mild septicemia, you get an inflammatory response against those agents, and then you clear in the mouth, which is sort of an interesting question. Yeah.
You can qPCR F nucleatum out of stool. There are a ton of papers that have qPCR primers for F nucleatum. So if you could get a stool sample or even an oral sample.
Yeah.
Probably
just oral,
Well, we have that, right? That's what Ryan has from the saliva, and then they have from the subgingival plaque. But what I should also tell you is for one of the field centers, we have tissue. We got a ton of polyps. We got all kinds of stuff on these people. My God, we could be looking at this, right?
Well, that's, you can totally sequence. Colon tissue is easy to sequence.
Now it's FFP. I should tell you, it's FFP. Yep.
Possible, but challenging. You'd have to do marker sequencing, so we couldn't sequence the whole genome. But you can fingerprint the bacteria in a way that doesn't pick up as much human tissue. And doing that fingerprinting technique, there's a lot of stuff you can potentially look at even in an untargeted way.
Let's all talk. And so for Noel, this work is with Dominique Michaud. Remember
Okay,
the piece we all
Harvard,
wrote?
yeah.
You remember? Well, she's at Tufts now, but do you remember from that piece that we did for clinical chemistry?
Yep.
Yeah, so she was one of the people.
Well, I know her work.
Remember?
Did she work with Edward Giovannucci at some point?
Mhm.
Early on, yeah. I know her cancer work. She does great stuff.
Yeah.
Yeah, and Liz, so that'll be interesting. You also have methylation on the ARIC study. So
We do.
once
Yes.
you do the
right.
sequence
Yeah.
of nucleatom and so on, it'll be nice to kind of correlate the methylation. It'll be cool.
What a good point. Okay, wait, we
Yeah.
don't have to
Yeah,
talk
you guys
again.
all have to get together.
Yeah. That sounds good. So Norma, that's the value of the coffee hours, right?
Yeah, this is one of the
That's
goals,
right.
to create new
Mhm.
collaborations and come up with new ideas. It's
Mhm.
fantastic. I'm so thrilled that this is happening. This is great.
So now we just have to make it happen, meaning get together.
Yep.
Yep. With a clear reason and plan, right? So that, because often people just get together, nothing happens. So we have to have like a target, right, for what we want to accomplish. All right.
That's always the nice thing about RFAs, right? They bring everyone together with a reason and plan, but you could do it outside of that too. We can make our own.
All right. Well, good. That is good.
Justine, I love the fact that you brought up that important difference in the mouse models, that they're coprophasic too. It's just like great to remember all these. That's a key difference, hopefully in humans, at least adult humans and mice, right? So hadn't thought about that, but that's true for the oral, or I guess also just they're kind of recycling their microbiome constantly, right? That's fascinating.
Phage effects are a huge confounder in your mouse studies, and they are obnoxious as heck. I also have a friend who sequenced the entire digestive tract of a mouse and did metabolomics all along the digestive tract. And one of the fun things was seeing the fecal community in the mouth and the fecal community out.
Wow. Whereas in humans, the mouth and the gut are not very close. But in mice, they probably are almost identical.
I mean, yeah, it's that recycling.
That's wild. All right. Well,
Time to
we are
wrap it
actually coming up on 6 o'clock here, and I think this has been just really great. This has been one of the better coffee hours we've had. A lot of really great discussion, and people are obviously enthusiastic. Some people came, some people went. But I hope you all feel that it was fruitful, or at least interesting. I got a lot out of it. I learned a ton. And
I love this. This is great. Thank you, Alan.
Yeah, sure. And you'll all get invited, obviously, everybody's invited to the next ones, and they're always hopefully interesting. Even
And
though
hopefully that is with beer and pizza.
Yeah.
Yeah.
I mean, literally, we got the email today that the masks are dropped.
Both of which will have impact on the microbiome and the metabolites.
Yeah.
Yeah.
So we just want to know, Alan, if the beer is good for your microbiome.
Well, I've been doing a very long-term longitudinal N of 1 study on that. And so far, at least I haven't detected it much untoward effects, let's say.
All
There's
right.
definitely
I've had minor
alcohol
memory
degrading
lapses.
bacteria.
Well,
I haven't
this
checked
is,
my microbiome.
I agree with Alan, and thanks so much to the speakers.
Yes,
for
thank you very
this
much, Hari,
session
for coming again.
today.
And Noel, it was,
Yeah.
again, a real
Yep.
pleasure to have you. And Justine, it's nice to meet you.
I just want to say also that, not only thanks for the invitation, but Hari and I were able to just get together before this, and that was
Great.
part of this whole experience for me, is just meeting with you, Hari. So thanks.
Fantastic.
Yeah, same here,
Yeah.
Noel. And it was fun interacting, and this whole interaction has been great.
Yeah, we really appreciate it, especially,
We have a
it's a
lot of
longer
new
meeting.
ideas here.
So-
We have a
so
lot of new
thanks for
ideas to follow up. So that's
contributing
great,
the time. It's great.
I-
This was a really
I-
good meeting. All
And
right, any other last thoughts before we close out?
Nothing for me.
Everybody's talked out.
Well, it was very productive in terms of the conversation, I think. So so glad you two could both get together and present together, and we could have this discussion.
Great.
Appreciate it.
Thanks, Norma, for arranging.
Sure. Good night,
Thank you,
everyone.
Nicole.
All right, take
All
care.
right, have a
Bye.
good one.
Bye.
All right, thanks
Bye.
everybody for hanging in there. See you all.